my name is Hwang, a PhD student at Songyungan University. I'm in Molecular Tumor Immunology Lab with Professor Chang Lee. Our lab mainly focuses on the novel pathogenesis and molecular targets for treating disease regulated by C72 and pelino-1. Today, I would like to introduce a study, the ubiquitin ligase pelino-1 targets C3 to regulate macrophagy-mediated inflammation and tumor development. UC and CD are the two forms of IBD characterized by chronic inflammation of the digestive tract leading to diarrhea, rectal bleeding, and abdominal pain. One of the most severe complications of IBD is the development of CAC. Continuous exposure of intestinal epithelial cells to pro-inflammatory stimuli and excessive cell damage with increased intestinal epithelial cells turnover resulted in genetic and immunological alteration, making IBD patients prone to developing CAC. Ubiquitination is now recognized as the key regulatory mechanism in the pathogenesis of various diseases. Ubiquitination involves a cascade of enzymatic reactions mediated by three types of enzymes. Ubiquitin can be attached to substrate in multiple ways. The fate of the protein is determined by whether a single ubiquitin or a polyubiquitin chain is added, and by which lysine residue on the ubiquitin is used for linkage. Therefore, ubiquitination plays a crucial role in regulating various aspects of protein function and shaping the outcome of immune responses. To understand the role of ubiquitination in immune responses, it is important to consider its interaction with receptor-mediated signaling. In innate immunity, activation of such signaling is often triggered by PAMP and DAMP. Recently, several ubiquitin ligase have been studied for their roles in regulating receptor-mediated signaling, intestinal barrier integrity, and innate immunity. However, to fully understand the function of ubiquitin ligase and the pathogenesis of intestinal inflammation, further investigation is still required. Next, I would like to introduce pelino-1 on ubiquitin ligase. Pelino-1 is expressed in most immune cells, and this protein are localized in both the cytosol and the nucleus of the cells. Pelino-1 has been reported to regulate inflammation and autoimmunity throughout various receptor-mediated signaling pathways, such as TLR, IL-1 receptor, and TCRs. Based on this, we aim to investigate the role of pelino-1 in colitis and colon cancer. First, since pelino-1 is known to be activated by PLRs such as TLRs, we hypothesize that macrophagy-derived pelino-1 may respond to PAMPs to accelerate immune responses and increase inflammatory mediators. To test this, we treated BMDMs with various TLR agonists. We observed that pelino-1 expression increased in response to most TLRs, especially LPS, suggesting a strong link between TLR signaling and pelino-1 induction. To further examine how pelino-1 expression changes during colitis, we induced colitis in mice using DSS in drinking water. In colon tissues from DSS-treated mice, we found that pelino-1 expression was elevated compared to mice that received normal water. To investigate the association between macrophages and pelino-1 in colitis, we performed IF staining. After DSS treatment, we observed increased pelino-1 expression and macrophages in the colon. Additionally, pelino-1 mRNA levels were significantly elevated in macrophages from DSS-treated mice compared to healthy controls. This result suggests a strong correlation between pelino-1 expression and intestinal macrophages and colonic inflammation. To further investigate the role of pelino-1 in macrophages, we generated myeloid cell-specific pelino-1 knockout mice. To examine the role of macrophage-specific pelino-1 in colitis, we induced colitis in both wild-type and pelino-1 knockout mice using DSS. According to the DAI, pelino-1 knockout mice showed significantly less susceptibility to DSS-induced colitis compared to wild-type. Histological analysis also revealed that wild-type mice exhibit severe colitis. However, pelino-1 knockout mice showed localized immune cell infiltration and mild mucosal erosion in certain regions of the clone. Importantly, F480 positive macrophage infiltration was markedly reduced in pelino-1 knockout mice compared to wild type after DSS treatment. This suggests that myeloid pelino-1 promotes macrophage recruitment during clonic inflammation. Consistently, inflammatory cytokines like TNF-alpha, IL-6, and IL-1 beta were also significantly lower in the clone of pelino-1 knockout mice. So this result indicated that myeloid pelino-1 contributes to macrophage recruitment and accumulation during inflammation. In CAC model, pelino-1 knockout mice showed higher survival rates, less body weight loss, and longer clones compared to wild type. Macroscopic images clearly show that both the size and number of tumors were reduced in pelino-1 knockout mice. We hypothesized that the phenotype observed in pelino-1 knockout mice could be due to reduced accumulation of macrophages in the cloning mucosa. As expected, F480 positive macrophage infiltration into tumors was significantly reduced in pelino-1 knockout mice compared to wild type. This finding suggests that pelino-1 knockout mice suppress CAC development and improve antitumor microenvironment. Given the link between macrophage infiltration and disease severity, we compared intestinal macrophage subset in wild-type and pelino-1 knockout mice. While wild-type mice showed increased CD11b positive, CX3CL1 intermediated migrating macrophages with inflammation. This subset was significantly reduced in pelino-1 knockout mice. In contrast, CD11b positive, CX3CL1 high resident macrophages remained unchanged between genotypes. To verify the effect of pelino-1 on macrophage migration, wound healing and migration assay were performed using BMDMs exposed to chemotaxic gradients of immune-related signals. The wild-type group showed a significant increase in migration in response to LPS, CCL5, and TGF-beta, whereas the pelino-1 knockout group exhibited markedly impaired migratory ability. This result indicated that pelino-1 plays a critical role in regulating macrophage migration. Moreover, pelino-1 knockout mice showed decreased expression of key cytokines and chemokines involved in macrophage migration during inflammation. Therefore, we investigated the effect of pelino-1 on macrophage migration and its subsequent role in the development of colitis and CAC. Notably, our observation revealed a correlation between pelino-1 and post-STAT3. To elucidate how pelino-1 regulates STAT3, we demonstrated a direct interaction between two proteins. Furthermore, pelino-1 showed a stronger binding affinity for phospho-STAT3 than for its hypophosphorylated form. Next, we confirmed that pelino-1 functions as an E3 ubiquitin ligase for STAT3, specifically mediating lysine 63 linked ubiquitination. Using intestinal macrophages, under both normal and DSS-treated conditions. We also verified statutory ubiquitination in vivo. Furthermore, we demonstrated that pelin-1 induced the ubiquitination of phosphostatury. Taken together, pelin-1's statutory pathway is crucial in the pathogenesis of colitis and CHC throughout the lysine-6-linked ubiquitination of statutory. For more detailed data and experimental design, please refer to our published paper.